Hello, and welcome to this lecture on homeostasis. So, this is the lecture which came up with no sound, and I'm replacing that now with this lecture. Hopefully, I'll do better this time. So, a little short explanation as to what happened. So, as I mentioned several times in my letters and emails to you, this is new to me. I'm learning all this technology in the last couple of weeks. And I had recorded this lecture and looked at it and realized, oh my God, no sound. So I went back and did it again. And then I uploaded what I thought was the one that I had recorded with sound, but apparently I hadn't done that. I recorded the one with no sound. And anyway, long story short, you ended up with a lecture with no sound. And so I have to do it over, and I'm doing it. So let's begin. As I mentioned at the very beginning, this is about homeostasis. And let me write that term down and then talk a little bit about it before we launch into more of the lecture or more of our notes. So, homeostasis. This term, homeostasis, is going to be the key concept that runs through this entire course, and in fact runs through not only this course, but 40B and 40C. When you talk about the human body in these courses that you're about to take, you are talking about homeostasis. The word may never come up. You may never hear that term, but you are always talking about it. So what I mean by that is as we go through these organ systems, the digestive tract, the nervous system, the cardiovascular system, urinary, whatever system you talk about, in some form or fashion, its purpose is to maintain this thing that we call homeostasis in the human body. And as you go through the chapters, every chapter will have a list of the functions of the organ system that you're reading about. And those functions are, in fact, the way that organ system helps to maintain homeostasis in your body. So this entire course, and I mean all 3 quarters, is all about homeostasis. So if there's going to be one key concept, one key principle or process that unites everything that you learn, it's this, homeostasis. So let's define this term homeostasis. And we'll begin with breaking down the word itself and seeing if that gives us any information. And so this word is composed of 2 terms, homeo, which means sameness, and stasis, which means standing still. Okay. So sameness, standing still. That kind of captures what this term is all about, but it doesn't really give you a good, strong definition, something that you can hold on to and use as a tool to understand what you're about to learn about the human body. So let's get a good definition of what this means. And homeostasis is going to be defined as the maintenance of nearly, and there's an R in there, static or constant conditions in the internal environment. Despite external and internal changes. So the maintenance of nearly static or constant conditions in the internal environment despite external and internal changes. So the key thing that I want you to focus in on, the key words on this is nearly and internal environment. Okay. So nearly, not constant, not right on the money, but nearly static or constant conditions in the internal environment, despite changes that are taking place either externally to your body or internally in your body. Okay? All organs and tissues are going to perform functions to help maintain homeostasis. And this is exactly what I had said earlier in the beginning on the introduction of this idea. Everything you study, all the organ systems that you look at, their purpose is to maintain nearly static internal conditions despite whatever may be happening around you in the external world or even what's happening inside you. So let's take a look at some of these conditions. What are they and what can we learn from them? So let's turn our attention to that. And I'm going to give you a list. This is a handout that you should have already, because I've asked you to hand out these, or to take these handouts and print them out. So here is important constituents and physical characteristics of extracellular fluid. We'll talk about what extracellular fluid is and how that is different from intracellular fluid, and we'll break those fluid compartments down in just a little while. But let's take a look at some of these characteristics, some of these constituents. So along this column you can see oxygen, carbon dioxide, sodium, potassium, calcium, chloride, bicarbonate, glucose, body temperature, acid, base. As I said, this is not a complete list, but it's an important list. Many of these things are very critical for your survival. And then along this axis, you see what the normal value is. And then here's a range. And this is where we talk about that nearly constant, not this all the time. It's this ranging between these numbers, generally speaking. Can they go lower or higher than this and you'll be okay? Yes. So over here in this column, you see the approximal short-term non-lethal limit. So there's your actual range before you die. But obviously, if you're at 10, you're going to be extremely sick. And if you're up here, well, I'm not sure what happens up there. I don't know if anybody's ever gotten there. I suppose so, since they have that as a limit. But I have no idea what happens at that point to you. Certainly, if you're at the low end of this range, you are in big trouble. And all of these constituents and conditions that you see here have a range. And your body is constantly trying to maintain these ranges. It's not aiming for that number. It's aiming for between 35 and 45 millimeters mercury pressure in the extracellular fluid. Just take a look at some of these. So look at body temperature, for example. So that's your body temperature that you're supposed to read if you are taking a thermometer reading. But this is still a normal range. It is very tightly controlled, as you can see. It goes from 98 to 98.8. The average is around 98.4. So very tightly controlled. Acid base, again, that's your normal value, but here is the range in which you're okay, 7.3 to 7.53. Now look at the approximate short-term non-lethal limits. We don't have a lot of space to fool around with in terms of this particular constituent in our body. This is very tightly controlled here because that range is rather small. And once you start going out of this range, you're going to, well, that's approximately short-term non-lethal limit. So once you go out of that range, then you can kiss yourself goodbye. So All of these have different ranges. Some of them are more tightly controlled than others. One of the things I'd like to point out, particularly with this acid base, when we talk about internal changes, this is something that is constantly changing. So your metabolic activities, just the normal metabolic reactions that take place in your body, are constantly producing hydrogen ions. So your body is constantly fighting to stay within this range, because you're constantly producing hydrogen ions, which is going to force that number lower than where it wants to be. And so your body has to have certain processes and control mechanisms in place to maintain this normal range. The more tightly a constituent or characteristic is controlled, generally speaking, the more control systems are involved in making sure that range is maintained. So this is just some examples of some constituents and physical characteristics and their ranges. So again, the emphasis here is not on trying to get a specific value all the time, but to stay within a range. Let's take glucose, and we're going to do a little graph of glucose. So the normal value is 85 milligrams per deciliter. Okay? But there is my normal range between 75 and 95. And that's a pretty sizable range. And that makes sense because when you eat a meal, that's going to really push that number into a higher range. And you want, of course, keep it within this range, but you want to have some slack in there because that meal is dumping lots of glucose into your body on a regular basis. And then when you don't have a meal and you're exercising and working, then glucose is being consumed at a very rapid rate, and so that would force it in the lower range. But you want to control that range. So let's take a look at how your body might be able to do that. So we'll take this and we'll exchange it for this, which is a graph. And again, this is something you should have. If you don't have it, go ahead and pause me right now and go and grab this. And what we're going to do is we need 3 colors. And we're going to go ahead and graph out changes in glucose. So again, this is the concentration in milligrams per deciliter. And here's my normal range from 75 to 895. And there is the mid-range, or the normal value as they call it. And so let's go ahead and see what happens over time to glucose. And now this is not going to be an exact, it's not based on a study or anything, but it is going to demonstrate how glucose can fluctuate and how we can control glucose within our body. So the first thing I'm going to do is I'm going to start right here, and I'm just going to do a gentle curve, and we're going to see this fluctuating over time. It might overshoot a little bit one way or the other, but it's going to basically stay between those lines. Okay, so that line represents the change in glucose over time in the extracellular fluid. Okay. As you can see, it's going to fluctuate over time. And the question is, why is it fluctuating? And if we can see, we got a high point here. So up to this point, it's increasing. And after this point, it's decreasing. And then it increases again and decreases. Why? The answer to that is really pretty simple. During this period of an increase in glucose, you have a meal in your gut. You've consumed some meal, you're processing that meal, and you're absorbing the glucose from the food that you've consumed. And so you have this big supply of glucose available. It's crossing the gut wall, it's entering into the blood. So glucose levels are going up because you're dumping a constant amount of glucose into the blood as you absorb the meal. Once the meal is gone, you're still going to be consuming glucose. But now you don't have this source of a meal anymore. So what you're going to have to rely on is what's in the blood to a certain extent. So as you start working, and no matter what you do, you're going to need glucose. Even if you're just sitting watching television, you're still going to consume glucose. And so over time, the levels of glucose are going to drop in your body. And so if we can graph that out here, then this would be no meal. Okay. Now, you'll notice that even with a meal, it doesn't go much higher than 95. That's milligrams per deciliter. And with no meal, it doesn't really drop too much below 75 milligrams per deciliter. Why doesn't it? You certainly don't have any ready source of glucose available in the gut because that's already been absorbed and it's no longer there. You're using up glucose. How come it doesn't drop, keep dropping and dropping and dropping and dropping? And the answer is you have control mechanisms in your body which are going to allow you to maintain this range. And now I'm going to put in two responses. And these responses are, and by the way, we do have a little key, so why don't we go ahead, pay attention to that before I forget. So the blue line is glucose. All right. So as I said, your body has a response to either an increasing amount of glucose or a decreasing amount of glucose. So I'm going to show you that response, and I want to tell you that the milligrams per deciliter does not apply to these. So the concentration of the two hormones I'm going to talk about, insulin and glucagon, are not reflective of these values. I'm putting them in there just so you can see the response of your body to these changing conditions. And so the first one I'm going to use is going to be insulin. Insulin is produced in the pancreas. So the pancreas, there are cells within the pancreas that can measure glucose levels in the blood. And as those glucose levels begin to rise, these cells respond by producing more insulin. And so what you would see, there would be a little bit of a delay, but you would see an increase in insulin that corresponds to an increase in the glucose. And now as glucose levels fall off, you're going to see a decrease. And then as you go along, it's going to increase again, and then it will fall off. So that is my response to increase concentrations of glucose in the body. And as you can see, the response of your body for an increase in glucose is an increase in insulin. And insulin, what insulin does is it has different effects on different cells. But for example, in the liver, it tells the liver cells to take that extra glucose and convert it into a molecule that we call glycogen. This is a storage molecule for energy that we have in the liver. And so we start taking that glucose out of circulation, out of the blood. And as we remove it, of course, glucose levels will start to fall. The same is true with other body cells. So insulin sending them a signal to increase the uptake of glucose. And when you put all those reactions together, you get a decrease in the amount of insulin that you find in the blood. Now, the next thing that happens, of course, is we get a drop-off in the amount of glucose, and that's not good either. So you have to have a response to that. And what you're going to see is an increase in another what you'll see is an increase in another hormone. This is called glycogen. And once again, glycogen is being produced by the pancreas, a different set of cells than the ones that produce insulin, but capable of measuring the concentration of glucose in the blood. And as they measure a decrease in the concentration, they produce a hormone called glycogen. And what glycogen does is it causes the liver, for example, to break down. I'm sorry, this is called glucagon. I'm getting those two terms mixed up. So the hormone is glucagon, and glucagon causes glycogen to be broken down in the liver and has some other responses in other parts of the body as well. So the overall effect is that you're going to get a response to increased levels of glucose and a response to decreased levels of glucose, all of which is going to help you maintain your glucose levels within the normal range. Okay? So that gives you a sense of homeostasis. And let's talk now. Let's apply that information to your bodies. And in order to do that, we need to take a step back and try to see the body and what it's composed of and how all of what we've just talked about relates to that composition. Okay? So, when we look at the human body, what we see is that there are approximately 75 to 100 trillion cells in the human body. Now that's a very large number. That's a very, very big number. There's a lot of cells. I don't know what graduate student counted those up, but some poor soul had to sit there for about like 100 years and count them. No, that's actually an estimate, obviously. So how big is that number? Well, I did a little bit of math, and just to give you an idea of the size of that number, try to relate it to something that you might be able to visualize. And so what I asked myself is, what is the total number of seconds in 1 year? Total number of seconds in 1 year, and that's this number right here, which is also a very large number, but not as large as the number we just talked about. Okay, so that's 31,540,000 seconds per year. Okay. And now what I'm going to do is I'm going to take $100 trillion and I'm going to ask myself, if I make $100 trillion in 1 year, how much money do I make in 1 second? So the answer is, by the way, this. So if you earn $100 trillion in 1 year, you would maybe be making $3,170,577 per second. And hopefully that gives you a sense of the problem we've got here. We've got a lot of cells, and each and every one of those cells needs something to be happy. So let's take a peek at that, and then we'll come back to this question about homeostasis. So we have all these cells, and each one of those cells are going to need to have certain things happen in order for them to survive and be healthy. And obviously, if your cells begin to die in your body, then you're in big trouble. So let's go ahead and take a peek at a typical cell and see how we are going to make that cell, or what that cell actually needs. So we're going to draw a big circle here, and I'm going to highlight that circle so we can see it better. All right. So there it is. This is our cell. It's a happy cell right now. And let's go ahead and bring in a nucleus. So that's the nucleus. Okay. Well, there's our cell. And typical cell in the human body. How do we keep that happy? Well, that cell needs certain things. So it needs coming in certain things to make that cell happy. What is that? Well, it needs oxygen, obviously. It needs amino acids to build proteins. That's what cells do, is they build proteins, among other things. Okay? It needs certain kinds of fats. It needs minerals. It needs a certain ion concentration. It needs vitamins. And it also needs signaling molecules, because this cell is not an isolated cell. It's part of a large body of cells, those 100 trillion cells that we've been talking about. And there needs to be communication. There needs to be coordination within your body. And one of the key ways in which that happens is certain cells produce what we call signaling molecules. They could be hormones, for example, which cause the change in behavior of other cells. And until that signal is received, that cell doesn't know what to do. So it's part of what is needed by this cell, going into the cell. If these things happen, if they are available, the cell is happy. But that's not the whole story, because we also need to take things out. It needs to get rid of certain things. For example, carbon dioxide. Carbon dioxide is a waste product, and we can't allow that to build up inside the cell, so we need to remove it. Hydrogen ions, we talked about this. Hydrogen ions, another waste product, always constantly being produced, needs to be dealt with. Urea, uric acid, so many different things, typically waste products. But also, remember those signaling molecules. Some cell has to produce those in order to affect the behavior of other cells. And so when a cell produces a signaling molecule, it has to be taken away and transferred or brought into contact with the cell that it wants to influence. Okay. So 100 trillion cells, all of them needing this to keep them happy. Now, this is basically a management problem. 100 trillion cells, all of them demanding certain things and all of them needing certain things. How do you manage that problem? And one of the things that I like to say about nature is that it is lazy. It's going to try to find the most efficient, least energy intensive solution to a problem. And this is through the process of evolution. So if you have two organisms that are solving the same problem in the environment, and one of them is using less energy to solve that problem than the other one, that one has an advantage. The one that uses less energy to solve that problem than the other one has an advantage. And over time, the one that is more efficient will survive longer and reproduce more. So we've got this problem. We need this cell to be happy. We've got to deliver certain things. We've got to take away certain things. How are we going to manage that problem? Well, what we're going to do is we're going to surround this cell with fluid, and that fluid is called the interstitial fluid. So one of the things that we will try to do then is to make sure that all of these things are either being delivered to the cell in the interstitial fluid or being taken away from these cells by the interstitial fluid. Okay. So if we focus on this fluid, then we don't have to focus on this. And by the way, the water that we find inside cells is called intracellular fluid. Okay. Right. So what we're trying to do is make sure that the intracellular fluid is happy. And the way we can do that is by making sure the interstitial fluid has everything that the intracellular fluid needs to be happy and to maintain homeostasis. All right. So that gives you kind of a picture of what our solution is going to be. But we need to look at this even closer than that because, as it turns out, there are many fluid compartments inside the human body. And when we look at typical amounts of fluid, then we see that each fluid compartment holds different amounts of fluid. So again, going back to the idea that we want to have the most efficient solution to this management problem, let's take a look at where that problem is. Excuse me, that's not the one I wanted to show you. Here it is. All right. So let's just take a look at men, and then we'll look at women. So here is a chart showing you where all the different body, or what all the different body water compartments are, and their approximate volume in percentage, and typical volume in liters. So we have total body water. So for males, it's approximately 60% of their body weight is actually water. So only 40% of your body weight, if you're a male, typical male, is actually a solid material. 60% is actually the fluid. And that amounts to 42 liters. So here again is that problem, that management problem. So one way we can approach this is to say we're going to make sure that 42 liters gets is controlled. So we're going to try to control 42 liters. But as you can imagine, that is not very efficient. That's a lot of fluid. And how are we going to manage 42 liters? So let's break that problem down into a smaller component, something that is a little bit more manageable. So let's take a look. Here we are, intracellular. Remember, that's the water we find inside cells. So that's 60% of total body weight. So that's 60% of that 60%. So that's about 25 liters. Okay, so we can subtract from 42 25 liters. And when we do that, we get this, 17 liters. Right? So I have taken now 42 liters, and I've managed that problem now down to 17 liters. Because remember, as long as I keep the extracellular fluid happy, the interstitial fluid happy, then the intracellular fluid is satisfied. So as long as this is controlled, then this is controlled, okay? So here we are coming back to our problem. So I'm down to now just 17 liters. That's the extracellular fluid. So that's what I'm trying to, that's my target right there, making sure it has enough oxygen and I take away the carbon dioxide and so forth and so on. But actually this is still a fairly large amount of fluid. And we're not as efficient as we want to be yet. So can we manage this problem down into something a little bit more manageable? Absolutely. So so the 17 liters includes interstitial fluid and blood plasma. And it's getting kind of late, and I've done a number of these today, so I'm a little bit whippy. But so this is part of that extracellular fluid. This is the interstitial fluid, and that's what we're seeing right here. So instead of doing 17 liters, I'm now going to focus on 13 liters. So that's a little bit more manageable. I went from 42 liters now down to 13 liters. So I'm still in pretty good shape there, but I'm still not where I want to be. I want to simplify this problem even more, and this is where I'm going to focus, right here. That 3 liters. That's what I want to focus on. And there's a good reason I want to focus on that, which I'll explain in just a little while. So if I control that 3 liters, everything else falls in place. If I control these 3 liters, then the interstitial fluid gets what is needed. And if this gets what's needed, then this gets what's needed, okay? And that ultimately satisfies this 42 liters. Now, you'll notice there's 1 liter of transcellular fluid. What is that? Transcellular fluid is specialized fluid. It's found in the eyes, synovial joint, cerebral spinal fluid. So it's really not part of this equation, although it is an important component of your body. But in terms of managing, keeping cells happy, these are the compartments that we're focused on up here. So I start off with 42 liters. I subtract 25 because that's inside the cells. If I keep those cells happy, I will be okay. I can do that by making this 13 liters have everything it needs, and that's good. But I can even simplify the problem more because I can take this 3 liters and focus entirely on 3 liters. So I go from 42 liters to 3 liters in terms of managing my problem. If you'll notice, for women, it's a slightly different picture, but there are some very strong similarities. First of all, you'll notice that the percent body weight is lower. That is, the certain body weight of water is lower. It's only 50% as compared to 60%. Why is that? And the answer to that question is this. Women tend to have more adipose tissue. And adipose tissue is composed of adipocytes. And this is a picture of an adipocyte. And what you find inside an adipocyte is a very large droplet of triglycerides, fats. So the cell is completely filled with a very large droplet of fat. It's contained within a membrane, but it takes up most of the space. Now you know, and I know, that when you put oil in water, the two don't mix. So if I have water in this cell, none of that is going to be found inside this big droplet of lipids. So the water is now confined to this very small space inside the cell. So if I have more of these cells, I have less fluid in total because these cells don't hold a lot of water. So that's where we get this. That's where we get this reduced percentage of total body weight in water. It's 50% because females typically have more adipose tissue. Why, you ask? The answer is quite simple. They have to produce new individuals. They carry a baby around for 9 months. That's a tremendous strain on the body. There's lots of energy that gets consumed to build a new individual, and so they have to have the ability to store extra energy in order to get that job done. But the one thing I do want you to notice here is this number right here. Notice that that number is the same as for a male. So females, even though they have a total body water less than 35 liters as opposed to 42 liters in male, they still have 3 liters of fluid in the blood plasma. So this is the fluid that's found in your blood vessels, in your heart, your arteries, your veins, and so forth. Same number, same volume, and there is a very good reason for that. It's because that 3 liters is the optimal amount of fluid that you can store inside the cardiovascular system that will get the job done of carrying all of the necessary ingredients to keep a healthy body and to take away waste. So over evolutionary time, this is the number that has evolved. 3 liters of fluid. This is not counting blood cells. So we can add another approximately 3 liters of blood cells, volume of blood cells to your total volume of blood. So again, this is blood plasma, which is the liquid phase of your blood, not the cells. All right, so that's how we manage that problem. Now, when we do that, we've got a good solution to our problem. And so coming back then to this chart breaks down that very thing that we looked at in a bar graph now. So we've got slightly different numbers. That's okay. You'll see a lot of variation. And remember, these are ranges. So there's a female around 55% according to your textbook. The source that I used was 50%. Doesn't matter, 5% one way or the other is not significant. 60% for males, so that's the same. And we're breaking that 60% down into intracellular fluid. That's 2/3 of that 60%. 1/3 is extracellular. So I'm trying to manage this right here, keep this happy. If I can keep this happy, if I can get this to be stable, if I can get this to stay in homeostatic ranges, that will stay in homeostatic range. But I can manage this problem even better than just focusing on that. I can manage it down to this 20% of that 1/3. That's my blood plasma. And as you can see, that's found in the circulatory system. And then I can supply the interstitial fluid, which surrounds the cells. Okay? So that gives you a sense of how all of that is put together. And in addition, just to be clear, what's going to happen now is we're going to deliver that material to the capillaries. And let's go ahead and finish this picture. I'm going to expand on this picture in a slightly different way. So if we take, and here we'll do this drawing. So we'll do a little drawing here to show you exactly how all of this fits together. So we're going to make ourselves a capillary bed. And you'll notice that I'm leaving little spaces in the capillary bed. And this is a very simplified capillary bed. And what we're going to see here is blood's going to come in. So this is fresh blood entering this end of the capillary bed. And the vessel that it comes into is called an arteriole. You'll learn about that in 40B. And it leaves the capillary bed in a vessel called a venule. But as it comes in, it's going to, fluid is going to leave that capillary bed. So you'll see fluid coming out. And it comes out because we have a lot of cells that are closely associated with this capillary bed. And all of those things that we listed earlier being delivered to those cells. And then what happens is the fluid, once it leaves, circulates around those cells, and then it's collected again. So we bring back now fluid. And that's how we remove waste and take those signaling molecules. Okay? So that's the basic design of your body. So these capillary beds are the place where we exchange materials between, this is the interstitial fluid, and the blood plasma. All right, so here's a little fun fact. So few cells are located more than 50 micrometers, and that's a very short distance, from a capillary bed. Okay? And what this does is it ensures diffusion of almost any substance within seconds to the body cells. So the takeaway here is the management of 100 trillion cells. How do we do that? Well, we broke that problem down and we've looked at the water compartments, and what we discovered is that we're going to focus on just a very small amount of fluid, the fluid that's found in the circulatory system. And the way the circulatory system is designed with the capillary bed and these very close cells, that system then allows us to deliver what is necessary for those cells to be healthy and to take away the things that keep those cells healthy. So one of the other things I'd like you to point out at this point is that this is a coordinated effort, isn't it? Your body is coordinated in the way it's put together and how it functions. So we're trying to maintain homeostasis. We're doing that by focusing on a small percentage of the fluid that your body's composed of, and we're putting that in a system that has the capacity to deliver the necessary things to cells within seconds of when it's needed. Okay? So it's actually, when you start thinking about it in this terms, it's a beautiful, beautiful system. It's an amazing system the way it's put together. So to reinforce what I just said, let's take a look at this chart right here. And as you can see, this is a schematic drawing that's supposed to represent your body. And in this drawing, we have three organ systems. We have the respiratory system, we have the digestive system, and we have the urinary system. And then we have this, which is so I guess that's actually four. That's the cardiovascular system. And we've got the interstitial fluid, and we have the cells. So this represents the capillaries, so that's our exchange. That's where exchange is taking place here, okay? So oxygen is leaving, carbon dioxide is coming in, and we're circulating that to the lungs. So we're going to dump the carbon dioxide, we're going to take on oxygen, then we pass that through the digestive tract, and that's where we get our nutrients, our ions, our minerals, our vitamins, our fats, our amino acids. So all that's being added right now. And then we can manage the waste products as it passes through the urinary system, so we can get rid of hydrogen ions and other things of that nature. So this is a coordinated system, and it's all based on being able to deliver these 3 liters to all these organ systems that are capable of modifying the contents of this 3 liters of fluid that we find in the cardiovascular system. So we have taken this huge problem, 100 million cells, and we've simplified it down to 3 liters of fluid, which is connected by a system to all the other organs in your body. And all of those organs are in some way contributing to the health of that 3 liters and thus the health of all those 100 trillion cells. Okay? So to summarize, and you should absolutely have this in your notes, blood plasma, which is the liquid part of blood, is the optimal fluid compartment to control homeostasis within the body. Okay? And for these 3 important reasons. Number 1, it is a small volume. Of fluid. Remember, that's 3 liters, not 42 liters. Okay? Small volume. Number 2, it's in a self-contained high-pressure system. And that is the circulatory system. And finally, it is sent to all body organs that can control or modify the blood plasma. Okay. So we've got this idea of homeostasis. We see what that means. It's the ability to maintain nearly stable internal conditions within the human body. When we look at the body in detail, we see that it's composed of all these trillions of cells. These cells are surrounded by a body of fluid, that is the interstitial fluid. And in order to keep the cells happy and in their homeostatic range, we're going to make sure that interstitial fluid has what is necessary to help those cells be healthy. And we're going to do that by controlling the blood plasma and found within the cardiovascular system. All right. And so, ladies and gentlemen, the rest of this course and the rest of what you learn in 40B and 40C is all about this. It's all about what we're talking about right here. You will see the details of how that happens for each of the organ systems that you study. But that's what's happening, is you're controlling those internal conditions in the way we have just outlined here in this part of the lecture. All right. So I think we have a good handle on that. What we want to do now is we're going to turn our attention slightly to, okay, so we've got to control all this. That's where we're going to control it in the blood plasma. But how do we coordinate that activity? How do we know when to do this or when to do that? That kind of thing. We have to have some kind of mechanisms built into our body that knows when things are going wrong and can make a change in that process. And that's what we're going to talk about right now. So this is the heading. It's going to be general characteristics of control mechanisms. Right. General characteristics of control mechanisms. So we're going to describe this in a general way, and then we'll get a little bit more specific about those general characteristics. Okay? So in order to control any variable, and I don't care what variable that is, if it's the room temperature, it doesn't matter, or if it's the pH of the blood, it doesn't matter. You need three components. You need three components in order to get this job done. If you're going to control a variable, you need three components. These components are, number one, a receptor. This is the terminology we're going to be using. A receptor. What is that? It's going to be some sensor. Okay? And this is going to be able to measure or monitor the variable. If you're going to control some variable, you've got to be able to measure it. You've got to know what it is at any one particular moment in time. So if you're talking about the room temperature, you have to be able to measure the room temperature if you want to control it. Right? Same thing, you've got to be able to measure the pH of your blood if you want to be able to control that. You've got to be able to measure the glucose concentration in your body in order to control it. So you have to be able to measure a variable, and that's going to be called a receptor. So I'm going to use an example, one that we haven't yet used, and that's going to be an example of arterial blood pressure. Now that wasn't on that list that we looked at earlier, so I'm bringing in another example. Arterial blood pressure. This is the pressure of blood inside arteries. Not on the venous side, not on the venous side, but inside arteries. Arterial blood pressure. Okay? So we can measure this in our body. And those things that measure that are called baroreceptors. And they're found in a couple of key areas in the body. They are found in the walls of the carotid artery. Number one. And number two, in the arch of the aorta. Now you're going to learn about both of those vessels when you get into 40B and you do the circulatory system. But very briefly, the carotid artery is a artery that runs through the neck. And when it reaches about this level right here, it splits into the external and internal carotid arteries. The internal carotid arteries supply blood to the inside of the skull. The external carotid supplies blood to the outside of the skull, all the face, tissues, and the muscles and the skin and so forth. But right where it splits, there's these baroreceptors and measuring the pressure inside that artery right there. The arch of the aorta is the major artery that comes out of the heart. It comes up, straight up, and then it does this big arch and drops down through the thoracic cavity and the abdominal cavity. And in that arch area, there is some more baroreceptors. And what they're doing is they're actually physically measuring the stretch of the wall of the artery. So if there's an increase in blood pressure, there's more stretching. That's measured, and that information is taken in. If there is a reduction in the pressure against the wall of the artery, then the artery tends to collapse a little bit, and that's also capable of being measured. So we're measuring the stretching or the collapsing of the artery walls right there. Now, and as a result, you're supposed to be able to respond to changes in blood pressure. So if you just think about this for a second, the positions where we have these measurements, or we can measure this blood pressure, right here, just before it goes into the brain, and right there at the heart, where that artery is going to supply blood to all the rest of the body, except for the heart, except for the lungs itself. Okay, so two key areas: brain, rest of the body. And you're measuring blood pressure there. Now, it's interesting. I think we've all had this experience where you stand up and you get dizzy, And what has happened there is that these baroreceptors in the carotid artery and the whole system that we're about to talk about hasn't responded fast enough to control blood pressure. And as a result, blood pressure wasn't controlled. Blood pressure actually physically dropped. Your brain was momentarily deprived of enough blood, and you got dizzy. Okay, and that's kind of interesting because it tells you how important blood is to your brain. Just a momentary change in blood pressure can cause a very big difference in the functioning of your brain. All right, so we measure then that variable, in this case, arterial blood pressure. What's next? Well, the second thing that we need then is we need to be able to control that variable. We need to have a central location where information is collected and decisions are being made. And so this is the second piece of our puzzle, and that's going to be called the control center. So we have a control center. And this, as I said, is going to integrate information. It's going to set the range for various values. So, for example, body temperature is set inside the control center, which would be your brain in that case. And it's going to get or receive input from the sensory receptors. Okay? And then it generates an output. Generates an output. All right, let's go back to our example of arterial blood pressure. Let's make this a little bit more concrete. So, example. Inside the brain, there's an area of the brain called the medulla oblongata. I'm going to shorten that to medulla. So the medulla of the brain is, inside the medulla is a area which is called the vasomotor center. And that is going to receive the inputs from the baroreceptor. So input from the baroreceptors. And then it can do an output. It produces the output. So it gets information, and then it produces an output, which can be either a decrease or an increase in the number of signals to the heart and blood vessels. So it's receiving information and generating an output based on the information that it's receiving. Okay. Now, the final piece of the puzzle. The final piece of the puzzle is going to be the effector. So we have input, we have integration, and then we have output, and something is going to respond or change that variable. Something has to control that variable. And that's the third piece of the puzzle, and we're going to call this the effector. Okay. And this is the structure that receives output and makes a change in the variable. Okay. It affects change. An example is going to be heart and blood vessels. So if we get a change in blood pressure, we can adjust for that change by either increasing or decreasing the heart rate, by constricting or relaxing blood vessels, depending on what direction that variable is changing, whether we're getting an increase in blood pressure or a decrease in blood pressure. Let's take a look at a schematic of what we just wrote down in our notes. So we'll have a visual image to relate to. So we have some stimulus disrupting homeostasis by increasing or decreasing that value. So we have a controlled condition, which is the value, that is being monitored. And it's being monitored by receptors. So that's our first bit of the puzzle, the receptors. Those receptors are then going to send the signal, that's the input, to the second part of our puzzle, which was the control center. This receives the input and provides for an output, and that output will go to the effector, and that will bring about a change in the value that we are measuring. So the response then alters the controlled condition, and we're going to return to some homeostatic condition. Okay, so that's basically what we've been talking about, and what we were able to put down in our notes in words. This whole thing, and this chart, by the way, is in fact in your textbook if you want to reference it. Let's take this a step further and apply it to the specific example that we were using as we wrote our notes. So it's going to be, as you see here, some stimulus disrupts homeostasis by increasing, in this case, increasing blood pressure. So there's an increase in blood pressure. The first thing we need to do is measure that. So that's our baroreceptors. And as I mentioned, those are found in the carotid artery and the aortic arch. That information is then going to be sent into the brain, the medulla oblongata, the vasomotor center. That's going to register that change, integrate that along with other information it might be receiving, and produce some output. And that's going to change heart rate and blood vessels. So there's going to be a decrease in heart rate, and a decrease in heart rate, and a increase in diameter of blood vessels. So the smooth muscles that surround arteries, certain arteries, going to relax a little bit and allow those arteries to expand a little bit, and that will help to reduce blood pressure. So we're going to get a two-fold response: a decrease in heart rate and an increase in diameter of blood vessels, and that's going to then return back to homeostasis. We're going to oppose the increase in blood pressure by those two mechanisms. Okay. All right. Well, that's all fine and dandy. Let's talk about the two kinds of control systems we have in our body. And we're almost done here, ladies and gentlemen. So just hang in. I certainly am trying to hang in here. So two kinds of control systems. We'll list them and then we'll talk about them. Number 1, negative feedback systems. Okay. Number 2, positive feedback systems. Okay. Let's focus on negative, and then we'll talk about positive feedback systems. So starting off with a negative feedback system. Okay. This is going to be the most common. Okay. And the way you tell if, so you're going to have to distinguish between a negative feedback system and a positive feedback system. How are you going to do that? And this is the key right here. What you're going to see is at the end of that system, do you get a reduction or oppose the original stimulus? So let's write that down. So it produces a result that reduces or opposes the stimulus and returns the value back towards its normal range. So a negative feedback system is the most common, and I think if you think about it for a little while, it makes sense. Because remember, we started off talking about trying to maintain all of these different variables within certain ranges. Glucose, oxygen, carbon dioxide, so forth. That's one of those first charts I showed you. So we had these normal ranges, and what we realized is that by having the external environment or internal environment change, those parameters, those values are trying to be pushed out of that normal range. So we want to keep it within that normal range. And this is what we're going to use is our negative feedback system, because as it moves out, it's going to be opposed. It'll bring it back in. Let's use the example that we just talked about, which is a blood pressure, and see if this is a positive or a negative feedback system. So here it is. So what you want to do if you're analyzing whether this is a positive or negative feedback system is to look at the initial stimulus. The initial stimulus in this case is an increase in blood pressure. Okay. Then we go boom, 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 all the way through the process. And what do we end up with? A decrease in blood pressure. So we go from an increase to a decrease. Is that a negative feedback system? Yes, because we're opposing the increase and with a decrease. That's a negative feedback system. Okay. Now, if we had an increase in blood pressure, and after we go through this bump, and we come out here and we get a further increase in blood pressure, then that would be a positive feedback system. Fortunately for us, those are fairly rare because there's very few times when we actually want that to happen. So a positive feedback system is quite different in that, let's get it down. Positive feedback system. Okay. This is going to tend to increase or reinforce the stimulus. Okay. These are rare, as you might imagine. But some examples of where this is necessary would be childbirth. That's a positive feedback system. Blood clotting. Orgasm. And urination. All of these are examples of positive feedback systems. Okay. Let's just focus on childbirth. So during childbirth, at some point, of course, the uterus begins to contract. And that is going to put pressure on the baby, forcing the baby, pushing the baby down through the birth canal. Now, as the baby moves through the birth canal, of course, the head and the body is going to stretch the walls of the birth canal. That's a signal that's being sent back into the brain. And there is an area in the brain that's going to release a hormone called oxytocin. And oxytocin will be released into the bloodstream, will travel down to the uterus and cause stronger contractions. So the baby now is experiencing stronger contractions and is being pushed further and further down through the birth canal. As it does so, there's more stretching, more signals back up to the brain, more oxytocin, more contractions, so forth. It's reinforcing the process of childbirth. It only stops after the child is finally out of the body of the woman and the stretching has ceased. Okay. All right, so that's a positive feedback system. And again, what you're going to look for is you're going to look at that initial signal, that initial stimulus, and look back at the end of that process. Are you going to increase it? If you're increasing it, that's a positive. If you're opposing it, that's a negative feedback system. Okay. Now, within the body, there are two major control organ systems. The first one, I think you probably have guessed it, nervous system. Okay. So this is going to control, for example, skeletal muscle, some glands, some smooth muscle, okay, cardiac muscle, okay, and the hallmark of the nervous system in terms of its response rate is that it is fast and rapid. It's a very quick way to respond to changing conditions. This is the nervous system. Okay. One interesting thing I want to point out at this point is think about all the tools that you have in the toolbox to change conditions, whether it's internal conditions or external conditions. And another way of saying this is think of all the tools you have in your toolbox that we used as humans to get to the moon, to build computers, to create art, ballet, music, all the stuff that we are so good at doing, and all the creative endeavors that we do. What tools do we have in our toolbox to actually accomplish those things? And there's only two: muscles and glands. We only have two tools in our toolbox, a lot of T's there, to control our environment. So if you think about everything that we're capable of doing, that's pretty amazing that we can do that with just two very simple tools: muscles and glands. All right. Number 3 is the endocrine system. All right. And so here we're going to be using a chemical called hormones. And you're going to define what a hormone is in 40C. Okay. And typically we're going to focus on metabolic functions. So the endocrine system tends to focus on metabolic activities within your body, but also smooth muscle. Okay. And growth and development, of course. And it tends to work on the long term, on a longer timeline than the nervous system does. There are some exceptions to that. So, for example, adrenaline is instantaneous, and that's part of the endocrine system. All right, so that really brings us to the end of this lecture. Again, I apologize for the mistake in posting a lecture that had no sound. So I have done this lecture now 3 times. So, yay. And I will see you next time. Good luck, and I will see you in a Zoom meeting in sometime in the near future. Thank you very much.